As we have seen in our previous module about the elemental composition of different organisms, in case of bacteria, in case of yeast, in case of filamentous fungi, and we have seen in that table that there is a percentage presence of different elements. So in this slide we can see that while talking about the medium formulation or a media formulation, so these are very important component of the medium. First one is water, and then is the carbon source and energy source. Then is a nitrogen source. So there are different compounds that contain the nitrogen compounds, vitamins and growth factors. Sometimes we have to go growth facilitators, growth enhancers, and we have already discussed about, because there are some microorganisms which can not produce some specific organic compounds. We have to add into that medium in case of vitamins, in case of other compounds. Minerals and trace elements. Sometimes we add the buffers, some compounds which act as the buffers, because there is a small change in the pH that can affect the growth either on the production of some specific production. Inducers, because sometimes some substance act as the inducers that is only needed for a specific kind of product. Precursors, because in order to produce some specific product we need some precursors, because they had to, that is only specific cases in the fermentation which we called as biotransformation fermentation. Chelators, sometimes when we use such medium which have the possibility of present some compounds, then unwanted compounds and heavy metals, then we have to use some chelators. Especially in case of strict anaerobic conditions, then we have to use some reducing agent as oxygenation chelators. And then sometimes we have to use some inhibitors. So antifoams, these are different component part of any medium. So while designing or formulating any medium, we have to keep in mind that water, carbon source, energy source, nitrogen source, vitamins, growth factors, minerals, trace element, buffers, inducers, precursors, chelators, inhibitors and antifoams. So all these factors should be given weightage. Their presence, their quality, their cost, so many that should be talked about. We should talk about these components in detail one by one in our later modules. So here in this slide, we have seen that carbon is very important. We will talk about the carbon source later on, but here we will discuss about the role of the carbon. Carbon has the dual role. One side, that carbon is the part of the building block. On the other hand, that carbon source also utilized for the energy cycle, that also give utilize in respiration. And we know that ultimate loss of the carbon by producing the carbon dioxide, if we go in detail the respiration equation. So the carbon requirement for the biomass production under aerobic condition may be estimated from the cellular yield coefficient determined by Y. So cellular yield coefficient that can be calculated just by this equation, that quantity of the cell dry matter produced, that how much is dry cell mass, and then divided by the quantity of carbon substrate utilized, that how much is carbon utilized, that how much is the biomass produced. So that is the cellular yield coefficient, Y. So an adequate supply of the carbon is essential for the product forming fermentation process. So the carbon is very critical. So the major part of any fermentation medium is a carbon. So you can see in this table that the cellular yield coefficient, while using these different carbon sources, when we use methane, N-alkane, methanol, ethanol, acetate, malate, glucose with reference to the molasses. So when we use the molasses, you can see in this that the cellular yield coefficient is 0.51. Highest cellular yield coefficient, that is only in case of when they utilize N-alkanes. So this is the cellular yield coefficient reported by Abbott and Clayman in 1973. That is actually that how much gram of biomass produced in dry weight by utilizing that 1 gram of the substrate consumed. So according to the values from the table, as in a previous slide, so the bacteria with the yield coefficient or a cellular yield coefficient for glucose is 0.5. So if we calculate by this equation, when we take that cellular yield coefficient is a 0.5, so if we want to produce that 0.5 gram cells per gram of the utilized, and if we see that if we want to produce more, then the glucose needed to obtain 30 gram per liter of the cell mass, then we can utilize that 60 gram of glucose is needed. So by knowing the cellular yield coefficient, we can add the carbon source into the medium accordingly. So by using this, we can see here that approximately 1 liter of the medium would also need to contain approximately 3 gram of nitrogen, 1 gram of phosphorus, 1 gram of potassium, and 0.3 gram of sulfur, and 0.1 gram of magnesium. This one is the basic need when we have to design a medium in which we want to produce that desired biomass. Just by utilizing the glucose, just by knowing that the cellular yield coefficient, that can help to design the medium formulation in any fermentation process. So the fermentation media and the cellular yield coefficient, that how relate with the carbon source, that is very critical. We will talk about in our later module in detail about the carbon sources and their choices.